Okay, Bismillahirrahmanirrahim. Assalamu alaikum and good morning, good afternoon and good evening, everybody who are joining us here in person, people who are joining us online from different time zones. I welcome you all to comstech's hybrid workshop on drug discovery and development. I would like to welcome, first of all, our speakers, Professor Dr. Paul Denning. Thank you very much, Professor, for joining us and for traveling all the way from UK for delivering this workshop and for other activities. I'm also extremely grateful to Professor Dr. M.K. Paul, who has joined us online. Professor Paul, thank you very much. We understand you were due to travel. However, due to some urgencies, you are here with us online. Thank you very much. Extremely grateful for your online presence. The theme of this workshop is to understand advances of drug structure biology and drug design, particularly in the age of AI. So we will begin today with a recitation from Holy Quran.
أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الرحمن علم القرآن خلق الإنسان علمه البيان. الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان. صدق الله العظيم.
Jazakallah. May I now request His Excellency Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General Comstech, Academician and Vice President of the World Academy of Sciences, South and Central Asia, for his welcome remarks.
Good morning, good afternoon, and good evening, ladies and gentlemen. We are in various time zones, and I'm glad that we already have about 40 over people online from different countries. Thank you for being with us this morning. I know that the Eid holiday season has not really gone away. We still feel that we are in the same season we want to remain in Eid season, right? So well, apparently today's seminar will bring you back to hard realities of life. And for that to happen, we have a very dear friend, Professor Paul Denning, and Professor M.K. Paul with us. Both are very distinguished individuals people with great vision and mission, and I see that Patrick is online as well. So this wonderful group of people are the partner for a great mission with comstech. Comstech, with its own network in 57 countries, trying to conduct various programs and initiatives, and one of the programs in which we are partnering with Durham University is on neglected tropical diseases. I'm sure that you appreciate that neglected tropical diseases are important and that very little attention has been paid in terms of understanding the science of these diseases, and as a result of it, to develop therapeutic interventions. You know, why it is important to have drugs in the first place? Because we all want to be healthy and to prevent diseases and all that. But you see, drug discovery and development paradigm has been largely distorted towards care and management. So the drugs which you see are for largely managing diseases, not for the real cure. I don't know whether you agree with me or not, but the fact is that the diseases which we all suffer, our elders suffer, such as diabetes or dementia or cardiovascular diseases or different kinds of pains and arthritis, they are only managed, not treated. Do you agree with me? So there is a need of developing a real cure for diseases. And there are so many diseases which do not really have any cure at all, any treatment at all. They call neglected diseases, they call rare diseases, you know, and nobody wants to work on them because either they are too rare or the population which are affected from these diseases are poor people, so they can't afford to have medicines. So not only that, for prevailing diseases for which medicines are available, they're not as effective as we would like them to be. They do not really treat the cause of the disease, but there are many diseases for which there is no cure in the first place. And also the whole area of prevention of diseases is also missing. You don't really find many people working towards prevention of disease. So drug discovery and development is extremely important. And today, two very distinguished speakers are going to talk about neglected tropical diseases. A very important area, and unfortunately, not many people are interested in these diseases, though they are very common, affecting literally hundreds of countries and billions of people worldwide. But since they are the most poorest of poor in the tropical and subtropical regions of the world, where affordability of medicine is very little, both signs of these diseases is not focused and thus therapeutic. So they are neglected in a true sense of negligence. And there are only few people, few pharmaceutical companies, some international NGOs, and to some extent WHO, they're trying to do something for that. And among them, this group of individuals who established global network on neglected tropical diseases, which was established back in 2018, is a very important one because they start first time working on, I mean, of course, there may be other efforts also, but as a consortium of 6 countries, 3 in South America, Uruguay, Argentina, please. Uruguay, Argentina, Brazil, and also India, Pakistan, and UK being the lead. And they developed this consortium of scientists to understand diseases at a molecular level. So if you identify what are the molecular basis of these parasitic diseases, then you may be able to address the diseases at a much better level. So it was a very important effort to understand the science behind all these diseases, a molecular basis of it. And that was the primary focus of it. I met this very distinguished group of individuals in 2018 with my first visit in Durham, and I was totally impressed. It's not that that they were getting a major grant from UK GCRF, the Ground Challenge Research Fund, as a major project, but most importantly, their vision and mission, and their capacity and willingness to work together with many countries. So this network of individuals from different countries, led by Paul later, is an excellent group, which beyond the funding is still survived. And to some extent, we continue to work together. Comstech is benefiting from from this legacy of this network, and we intend to invite them to different member states. So they were in countries like Kenya, Ethiopia, Morocco. They were planning to go to Comoros. They were in Pakistan before, and so in different places, we spread the message of continue to work on neglected tropical diseases because pharmaceutical companies largely do not want to work on them. So you need to work on them too, because scientists, young people need to work on these projects and try to understand what they can. So with this, I would like to invite Paul and later MK. What is your MK? Paul is a wonderful dear friend. He was all set to come, but because of a family tragedy, he wasn't able to travel. And he played a very important role, central role in developing this network, which we all being part of it, and also the science which has been conducted. MK by profession is a structural biologist with a profound understanding of use of X-ray and developing better understanding of the molecules which are involved. So he worked as a part of a larger team, and he has been instrumental in developing better understanding of diseases such as leishmaniasis and Chagas disease. And without taking further time, I would like to invite Professor M.K. Paul for his presentation. Thank you very much. Thank you.
Thank you, Iqbal. Can you all hear me?
Professor, please share your slides.
Yes, can you hear me?
Yes.
Good.
So just for everybody's information, the title of Professor Dr. M.K. Paul's talk, the first talk is Structural Biology and Drug Design in Age of AI, Artificial Intelligence. Professor Dr. M.K. Paul is co-director and Durham lead for Engineering and Physical Sciences Research Council, Molecular Sciences for Medicine, Center for Doctoral Training. He is, of course, associated with the University of Durham. Professor Paul, thank you very much for joining us, and please start your lecture. We can see your slides.
All right. Well, thank you all for having me. And again, I need to start by apologizing for not being there. As Iqbal said, there was a family emergency. So I'm actually right now in my brother's house in his workplace. You can see all his books in the background. But I'm really happy that you managed to organize a virtual meeting. So I'm really pleased and I'm very grateful that I can give a talk on my work. Now, I'm going to talk about AI because this is basically everywhere on the news. And what I want to do in just about half an hour to give you sort of an uptake on what we can do with AI in structural biology and drug design. And I often call this the dangers and pitfalls of AI rather than the chances of AI. So most of you have probably heard about the structure prediction revolution, which really came out of something that's called the critical assessment of protein structure prediction, or CASP competition, which started in 1994 as an opportunity for really an independent assessment of protein structure prediction packages. And really cannot underestimate how much influence this had on the whole field. Because basically every couple of years, new structures were given to structure prediction people that had not been published, and they could basically fine-tune and hone their methods on these new structures, and then there's a bit of competition. I should say that the very early versions included secondary structure predictions and intrinsically disordered regions predictions. So these are tools that we have been using for quite a long time. The key part in terms of structure prediction that we need to distinguish is homology modeling, where we actually have a structure in the database that is somewhat similar. So there's a sequence identity between my novel structure and structure already in the protein database. But as I've written here, the holy grail was always to predict a structure that has absolutely no sequence identity with anything in the database and could be a new fold, a completely new fold, or completely new function. So in 2018 is really the year where things changed when AlphaFold came onto the scene. This is actually a page from the BBC webpage at that time where they said AI breakthrough could spark a medical revolution. In typical newspaper speak, this was already a revolution. I wouldn't say it was quite a revolution, but it certainly changed the field quite a bit, because this was for the first time that Google came into the play and used more AI methods in order for structure predictions. And for us as X-ray crystallographers, these structures for the first time, these predicted structure, were good enough to use them as a molecular replacement. So they were close enough to the real structure that we can use them to solve our X-ray crystal structures. So one step forward is CAS14 2020, which I would say was the real breakthrough, which was called AlphaFold 2, which was completely reprogrammed, and it used more and more of these deep learning techniques. And in the same time, Rosetta Fold was published from the Baker Lab, David Baker in Seattle, and they really took the ideas of AlphaFold 1 and programmed them into their own code. And I should say that, of course, Google or DeepMind had all the computational power of Google in the background. The Baker Lab in Seattle had all the servers for Microsoft. So in terms of computational power, they basically had unlimited power, or the most powerful computers on the planet at their disposal to develop this. And it's fair to say that both basically solved the simple protein prediction problem empirically. So they used deep learning techniques to predict structure of folds that were completely novel. And I've shown just one example up here from the paper, from John Jumper's paper in 2021, where the experimental structure is shown in green and the predicted structure in blue. And you can see there's basically a very good superposition. So the root mean square deviation of less than 1 angstrom is sort of within the experimental accuracy of your structure determination. And what was quite impressive for structural biologists is that even metal sites here, the metal site of this structure and the side chains of this metal sites were extremely well predicted. So clearly a breakthrough in our field in terms of structural biology. Oh, hang on. I'm not going to go too much over the algorithm. Everybody is, of course, invited to read the paper. But what I wanted to point out in this slide is basically the two inputs that these programs use. So one is this genetic database. So what the program uses is a huge number of sequences from the databases. So this is a multi-sequence alignment. So that means that the more genes that have homologous to our target gene are in the database, the better. And of course, the next generation sequencing has led to the position that we do have hundreds, if not thousands, of sequences that are evolutionary related. And this is actually quite a lot of information that is being used. And the second part that both programs use is the structure database. So if there's anything that is sort of close in terms of structure to our target structure, then this is also used as input. So what we really need to remember is that it is both all the sequences in the genetic databases plus all the sequences in the protein database and the structure databases that is being then used and put into the deep learning algorithm. And this works surprisingly well, and I would say surprisingly well, because our structural universe is actually relatively small. So if you look at the number of structures or the number of different folds, in fact, we structural biologists are always disappointed that there are relatively few different folds. So nature sort of always have evolved to come back to the same sort of basic scaffold, and then there's little changes left and right, few changes in the active site that then are responsible for the exact functionality. On the other hand, the genetic space is much, much larger. So that means that structure is more conserved in evolution than sequences. And that really is sort of the key for these programs to work. So again, a lot of data in the genetic database, not that much data, but quite a small set of folds in the structure database. Some really clever algorithms here in the evoformer and the structure model that I really admit I don't quite understand. But then in the end, there's also a bit of modeling, which uses physics-based modeling of existing programs to fine-tune the structure and make it even better. So let's go here. And of course, then you're all aware that this was awarded by the Nobel Prize, Chemistry Nobel Prize in 2024 to David Baker from the University of Washington here on the left-hand side. And he was actually awarded a Nobel Prize for the design of novel proteins. He designed the first new fold back in 2003. And then Demis Hassabis and John Jumper from Google, who headed the DeepMind protein structure prediction program. And at this point, I just want to show this picture up here. This is the Institute for Protein Design from David Baker's lab, because a couple of times I was asked that, why am I not doing the same thing? And then I always point onto this picture up here and say, this is the David Baker empire, where he has 150 people working for him or with him, developing these methods. So there's quite a bit of competition. I also always point out that there are certainly other people that might have been included in the Nobel Prize. And in fact, it was David Baker who said that he expected he would share the Nobel Prize with Stephen Mayo on the left-hand side and Bill DeGrado, who were early pioneers in protein structure prediction. And the reason I point this out is twofold. One, Stephen Mayo is actually quite an impressive person. He's actually the first Black professor at Caltech when he became professor in Caltech out of 400 faculty. So Bill DeGrado has done really crucial work on the prediction of helices. And I just want to sort of point this out that when AlphaFold came onto the scene, it didn't sort of burst on the scene of nothing. It really built on decades and decades of work of academics that they could use and build on. So important to remember, it's not just one genius, it's decades and decades of work that led to AlphaFold. So where we are with AlphaFold and Rosetta Fold today, we would certainly say that both have revolutionized the field of structural biology. And I'll just give you one example. This is actually a structure that we solved a couple of years ago in a very tedious process of multi-anomalous dispersion structure solution. So our structure, the experimental structure, is shown in blue. And then while we were working on this, AlphaFold and Rosetta Fold came out, and we used both of these and to predict the structure. And even though this has absolutely no sequence identity with anything in the database, both the red and the red and the orange fit quite well. So you can see that most of these helices fit very well. And it's just a couple of loops and the end termini where the structure prediction programs are really not predicting the structure. So the core of the protein, the basic fold, was predicted within about 1 angstrom of RMSD. So again, very, very impressive. And we could have used the AlphaFold models or the Rosetta Fold models to solve the structure by molecular replacement. But I think the first caveat is here that the structure prediction really missed two important things. First of all, they missed the dynamics. Now, this is just a little animation of a protein that is moving around. It's a different protein, but these are actually domain movements of these proteins that can be seen both experimentally, but also in computer models. And these dynamics are really not in the structure prediction. And obviously, the dynamics of the proteins in many cases are important for its function as well. But likewise, the structure prediction also misses the function. So we might be able to deduce the function from a structure prediction, but I would always say that really unraveling the molecular and in particular the cellular function from a structure prediction program still requires any hard graft in the laboratory. So we always come back that we actually need experimental science to validate, experimental science to use the information from structure prediction programs. So one more example that I want to give you about the function prediction. This is an example that, of course, Paul Denny is very familiar with because this is one of his favorite proteins. He might tell you more about this, the ceramide synthase. So I just want to show you the structure prediction and both comparing Rosetta fold versus alpha fold up here. And we can see that these 7 transmembrane helices up here in blue, they're very well predicted. And in fact, both programs predict these alpha helices really well. And the putative binding active site residues here in red, they're also predicted at a region where we would expect them. So in principle, right, we get an idea that, of course, this is a transmembrane protein. We knew that even before AlphaFold and Rosetta fold came out, and most of these proteins have 7 transmembrane helices. Again, it's a ceramide synthase, so it's likely that the active site is somewhere here in the center of the protein. But at the same time, the loop regions up here and the regions on both sides of the membrane are really not very well predicted. And in fact, the predictions of the 2 programs differ quite significantly down here. And while the active site is here and probably highly conserved, the actual function of this protein and how substrates get in and get out really depends on the loop regions as well. And the structure predictions in these regions are simply not very good and predicted at relatively low confidence range. So again, our structure prediction gives us a really nice picture of this transmembrane protein, but it really doesn't tell us all too much for its function. And this comes back to, I think, a recurring theme in my talk, that you really have to do the work in the lab in order to unravel the mechanism and the function. Okay. I give you another one example that actually also worked well, and again, an example that Paul is very familiar with. This is the SPT protein, where we do actually have a bacterial homologue in the database. So from the Toxoplasma gondii SPT, there's a bacterial homologue structure solved with a sequence identity of about 20%. And what we can see is that even in old problems like Swiss model, this catalytic domain up here was modeled extremely well. Because again, the structure is evolutionary more highly conserved than the sequence, right? Now we did actually this some time ago, and based on the Swiss model, we could see that the N-terminus down here is probably transmembrane helices. So we basically cut this off and made a domain with only the catalytic domain. But this basically never worked really well in terms of making a protein. So what we actually then could see is that with the AlphaFold model, we basically saw this additional helix up here. So this is a helix which is sort of elongated and not present in the Swiss model because the bacterial homologue doesn't have this helix. And AlphaFold was basically able to predict this helix, and sort of from the structure, we could see that the whole thing up here should be the catalytic domain. So basically just extending our construct just a little bit and cutting out all of these helices down here, these transmembrane helices, we could then get a much better expression, right? This was also supported by a model which actually gives you an idea about the conservation. So this model is then colored according to the conservation and the degree. And we can see that this catalytic domain is very highly conserved, but including this helix is quite highly conserved. And then it's less conserved down here. So taking this together, we then make a new construct and get all for the 7 much better expression levels and so on. I should say in terms of structural biology, this was still not enough to get crystals, so we still have not solved the crystal structure because this protein is still not behaving well enough in crystallization. So one of the things that not just we are, a lot of people are doing at the moment is using those AlphaFold models to improve their constructs for protein expression. And that's probably one of the most useful tools for people in the molecular biology lab. So, but then of course, how does AlphaFold work with IDPs or intrinsically disordered protein structures? And again, this is one of my favorite structures from the database. This is a human protein that I just stumbled upon, which is called SET binding protein. And it's responsible for a very rare but very severe neurodevelopmental disease called Schinzel-Gideon syndrome. Now, this protein consists of over 100,000 residues, and just the initial structure prediction says that it's almost entirely disordered. So when we chunk this into AlphaFold, we get something like this. And I let you think about this just for a little while. This is actually color coded in a way that the red is very highly confidence level and blue is low confidence level. So basically the program predicts a couple of helixes in the middle of the protein, and everything else is sort of completely intrinsically disordered and has no secondary structure. And even though the structure looks sort of globular and reasonable, because the protein in the end tries to make the structure more globular, of course, this model is not useful for anything. And we really can't deduce anything about the structure or the function of this protein. Now, the question really is this protein really completely disordered, or are the predictions simply very poor because there's very little sequence information and no structural information about those sequences of the protein? And that's something we sort of really don't know at the moment. But I must say, I find it extremely unlikely that a protein of 1,000 residues is completely intrinsically disordered and has no secondary structure whatsoever. So my hunch is, and I'm being recorded here, my hunch is that if anybody ever solves part of the structure, at least part of these molecules will be structured in one or the other way. So obviously, a lot of people have done then analysis of the power of AlphaFold, and I'll just give you a couple of pictures from a really good article from Agarwal et al. In Nature Chemical Biology, where they looked at different models and just sort of warning signs on different predicted structures. So starting with a peptide up here, this is a peptide superimposed with the PDB file, and you see this simply doesn't fit very well. So often on small peptides or relatively short stretches, AlphaFold doesn't seem to predict very well. And well, we can speculate about the reasons. Again, it could be that the sequence is not very highly conserved or very diverse, but it could just be that this is too small to bring together. This is another globular protein of insulin where, again, the structure prediction really doesn't quite fit into to the PDB file. Another examples are cofactors and metaproteins that lead to domain changes. So in this case, AlphaFold has predicted the helices really well, but the orientation of the helices with respect to each other really didn't fit quite well. And when we come to bigger structures, we can see that entirely domains could be completely different. So we could have domain movements with respect to each other that really, again, don't fit well. So basically, these are sort of examples, and of course, these are extreme examples that the authors selected from a large set of very good examples, but it's just as a warning sign. So AlphaFold and Rosetta Fold give you extremely good predictions in many cases, but in some cases, it is actually quite poor. So AlphaFold then is also being used for protein complex predictions, and that becomes, of course, important in the terms of cellular function, because most proteins, if not all proteins, do work in concert with other proteins. This is just a picture again from a review article how this is being used. So basically, we have the two proteins up here. We have two sets of multi-sequence alignments. We put them into a deep learning model, predict the structures, and then based on the structures, the program does basically a contact map. So it looks at sequences and amino acids that are likely to be close to each other, and from that, it puts them into a complex. So these context maps give you an idea where you have context between the two proteins. And that gives you an idea how a protein complex could be formed. And again, I'm going to show you a number of examples of good and bad where this works actually really well, and some examples where this doesn't really work well. But keep in mind, that really is important when we think about cellular functionality. So again, this is a similar picture of a larger protein complex, in different representations where we have the multi-sequence alignments up here. We have a bit of structure database. We put them into our deep learning model and do get structure modules. And this is an example of a structure that has multiple proteins, how they're being bound to each other. And again, there's of course some loop regions that are really not predicted well at all. But again, the protein is very good to give you low confidence levels. The individual domains are very well predicted with long helices, and this is mainly beta sheets. And from our context predictions, we can actually sort of see where these proteins should be linked to each other. Now, I must admit, I don't know how well this fits actually to the model. I'm pretty sure there's no structure of this protein itself. But again, it can give you a very important clue on where the proteins or two proteins can come together. And this is an example actually from our work that where we have used this. This is a protein that's called cellular retinoic acid binding protein that for completely different reasons we have been working on for quite some time. And our collaborators in Newcastle basically suggested that this binds to the cyclin D3, which in turn regulates a kinase so important for cancer, right? So they basically found, okay, there's this connection between retinoic acid binding protein that we have been working on with this cyclin CDK complex that they have been working on for a long time. Now, we didn't even try to crystallize this because it was very clear that this complex is very likely to be very transient and not really a strong complex. So we really then used AlphaFold to see if we can get an idea how these two proteins actually fit together, right? And this is the model from AlphaFold. Again, it's color-coded in terms of confidence level. And of course, the different proteins, so here's this D3-CDK4, and this one is our CRAB-PPP. They're extremely well predicted. And of course, that's to be expected because these are actually has been solved by several crystal structures. So there's a really good model up there. But what was quite good is that the interaction interface was actually very well defined. So on this, we could really see what is the most likely part of where these two proteins come together. And based on the AlphaFold prediction, we could then make, or the guys in Newcastle could then make predictions which residues to change in order to disrupt this interaction, right? Oops, this one up here. And I must say that the nice thing about this is that we really didn't give the person, the postdoc who did this AlphaFold modeling, any clue on how this could bind. And they really didn't know sort of which residues could have been affected. So we just gave them the sequences. They made the model. We looked at the model, and then it fitted really well to our predictions on and our mutations. So again, AlphaFold is a really good tool to test an hypothesis or to come up with an hypothesis. But the experimental validation still needs to be in the lab. So again, that's an example that worked really well. At the same time, we were also working with another group on more transient complexes, and this is something that a lot of people have probably come across. These are these frizzled transmembrane proteins that bind to Wnt domains to regulate development. So these are really important proteins in development. And what we basically did is we put this also into AlphaFold up here, and this is sort of where the model comes into place. And what basically happens is that the individual domains up here, you can see the frizzled protein up here and here, and our Wnt domain down here. These individual domains really don't fit at all. The individual domains really fit very well. So this is again our transmembrane proteins. These are the proteins on the solute side. But this protein down here should really bind on that side. So basically, the complex is completely wrong, and sort of all biological data really didn't fit. So the protein prediction really don't work in terms of getting the complex correct. We tried many different combinations and sort of tried to hone AlphaFold into getting this complex correct, but didn't. So this is a structure of the the domains by itself, and you see that this sort of horseshoe-shaped protein should bind this little protein down here. So from that one, we really couldn't answer many or any biological questions. So my point for this is that, again, AlphaFold needs to be always treated with a bit of caution, and we need to be able to have an experimental validation for any AlphaFold predictions. So moving tack a bit, and I'm coming sort of to an end, is where is deep learning and structure-based drug design? Now, structure-based drug design or computer-aided drug design really has embraced deep learning methods for decades. So if you do a little Google search, you see these terms in structure-based drug design actually decades ago. But what really did not happen in structure-based drug design is that we had this sort of eureka moment that changed the process, just like I've just explained for AlphaFold or for ChatGPT or DeepSeek. So deep learning techniques have been used for a long time, will be used in the future, but there certainly so far has not been the one breakthrough that can change the field completely, right? But on the other hand, because of all the hype, there's now, I mean, huge financial investment for companies. And you might have heard of some of these companies in Silicon Medicine. They've just made a $400 million deal. Accenture has moved with another company for another 600 million. Isomorphic Labs, that's the company that's been founded by the deep mind people and so on. So they have hundreds of millions, have raised hundreds of millions of dollars or pounds. So many of these companies that are springing up, and I do expect there will be a breakthrough coming relatively soon. So where does this sort of come into place for drug discovery? So the first thing is that when we talk about molecules, rather than proteins that are represented by just a string of amino acids, proteins can be a lot more complicated. Molecules can be a lot more complicated. So drug molecules can be represented by graphs, by smile springs, by volume with electrostatic surfaces. So again, compared to a protein structure prediction problem, dealing with molecules is actually a lot more complicated, right? Because our chemical space is many, many orders of magnitudes larger, and it's largely unexplored space. So again, for protein structure prediction, we have a huge genetic database, which is just one-dimensional information, and we have a relatively small contained structural universe where the structures always seem to be sort of the same. And that's really different for the chemical space, where, again, there's many orders of magnitude more possibilities to make molecules. And most of these is completely unexplored. So we really don't know sort of how they look like. And of course, in addition, when it comes to in vivo interactions, this adds another layers upon layers of complexity, how molecules behave in the body, how molecules behave in different parts of the bodies, and how molecules move from one part in the body and the other part. So I would argue that protein structure prediction is, compared to structure-based drug design, actually an easy problem. Now, nevertheless, people really, of course, use predictive structures now for structure-based drug design. And there's been a number of studies where predicted structures were compared with experimental structure in structure-based drug design. And I think one can summarize that if we have a set of well-defined structures from the PDB, for example, kinases, then the results are quite the same. And it's really difficult to say they're not really better, but they're also sort of in the same range. And that makes perfect sense because the predicted structures are based on the experimental structures, so they won't be far too very different. Looking at unknown structures, the results are somewhat mixed, I would say. There are reports that say that predicted structures work quite well, but then there's certainly quite a few papers out that I've seen where predicted structures were used, and even a very simple look with a bit of chemical knowledge onto their binding pose, where they'd put a small molecule into a binding site, tells you that that's not quite correct. AlphaFold 3 now has an option to do binding poses, predict a binding pose for a molecule. And in the first sets, AlphaFold 3 did not outperform the classical physical-based approaches in competitions. So far, certainly, it has not been the breakthrough in terms of deep learning methods, right? One aspect where these methods probably do work in the future are post-generation. So in principle, in sort of scoring the different binding poses, the different binding modes of a ligand, the potential binding modes, basically we used physics-based functions, and these really can be replaced and enhanced by nonlinear machine learning functions. And one of the programs that's out there, DeepDoc, at least claims in a nice paper, Nature Machine Intelligence, that is superior and in particular gives faster results. And it sort of gives you an idea how these programs work. So they represent our protein basically as a mesh with the binding pocket having 4 different properties: electrostatics, hydropathy, hydrogen bond donor, and shape index. And our ligand is simply represented by 2D representation. And the network is then learned on the basis of structures from the database, which is then applied to any new structure. I personally haven't used DeepDoc, but again, this paper just came out a couple of years ago, at least claims to be superior, and is certainly something that we will be looking at and comparing results with classical docking methods, right? In terms of new molecule generation, so this is the hit to lead generation. There are really quite a lot of AI and ML tools around, and they've been used for quite a long time, for example, to optimize pharmacokinetics optimization, improve ADME properties, fragment growing, crossing of blood-brain barrier, toxicity, and so on. And these have been really around for a long time. But I would say so far, none of these tools consistently outperforms the classical tools, particularly combined with human neural networks. So this is something that when I talk to people from companies, I always hear that people, particular group of people coming together, let's say from the chemistry and from the structural biology side or from the medicinal chemistry side, and brainstorming how the next molecule should be improved, still works better than any automatic tool. But I do expect that this is where the companies are most likely to make some advances. So future challenges remain. PROTACs, for example, these proteolysis targeting chimera proteins. In this case, you not only have to optimize your two ligands, your E3 ligase and your target protein, but you also have to optimize your linker molecule. And while E3 ligase ligands are usually well known, the protein of interest is novel. And of course, we know that the linker design is critical for in vivo efficacy. And I, again, know that people are trying to look into machine learning methods for that. But of course, the problem is with just a few hundred PROTACs molecules that have been developed and a handful being in clinical trials, there's simply not yet the database that can be used for deep learning. The situation might change once we have 10,000s of molecules that have been tested, and we know the few hundreds that might work, and maybe a few in the clinic. Nevertheless, there's an article I found very recently: AI and machine learning methods will be the future. It's very clear that each of the top 20 pharma companies have AI programs, and they often partner with what we call AI native biotech companies, so biotech companies that were founded on AI methods. And this is an example up here on sort of what's happening in terms of molecules that came from AI native biotech. So this is a relatively limited statistic, but it gives you an idea that since 2015, 75 compounds entered into clinical trials and 67 are still in clinical trials. Now, in this article that was actually published by Boston Consulting, there's one molecule up here that has been launched based on AI. Unfortunately, I couldn't quite find out which molecule this is. But we can clearly see that over the last 10 years, there's an almost exponential increase of molecules that at least are claiming to come out of AI methods. And there's no reason to believe that this is not going to grow further exponentially, right? Now, currently, small molecules seem to dominate the field. About 30% are small molecules down here. But biologics, such as AI-designed vaccines, nanobodies, and antibodies are on the rise. And I don't have time for this today, but Many people believe, I agree that these new diffusion methods for protein design that are used to design vaccines, nanobodies, and antibodies, they are really coming out at the moment very, very strongly, and they could well be the game changer. And I think the reason is that protein design, again, is actually easier compared to small molecule design because we are, again, dealing with a relatively limited protein structure data universe. So, in summary, and I hope I went into time, protein structure prediction itself has been an absolute game changer for us in structural biology. And we are indeed very grateful for this tool from AlphaFold and Rosetta Fold because of the power in terms of helping us in solving structures, but also helping us in coming up with new ideas and hypotheses. Right? These protein designed by AI or machine learning or diffusion methods, this is already creating biologics. There's a really impressive article 2025 from the Baker lab where they designed a molecule against a snake venom, so a protein that neutralizes snake venom. A really impressive paper by them. But also, and this really comes back to what Iqbal said in the end, the discovery of new therapies for those conditions that have no clear target. And these include neurodegenerative diseases, but also many NTDs where we simply don't even have a clear target. They are clearly beyond current methods in AI and ML. And that also comes back to what I've said before. Finding new therapies for those diseases will require hard lab work, and no computer program will just come up with a magic wand and give us a new therapy. And with that, I think I'm at the end. So hopefully I'm hopefully within time, and I'm very happy to answer any questions. Thank you so much.
Thank you very much, Professor Paul, for a very fascinating talk, I must say. With your permission and with permission of Professor Iqbal and Professor Denny, we are open for question and answers. I would request first our in-person audience here. Please raise your hand if you have questions. Kindly introduce yourself before your question. And also we have more than 120 almost people online. You're also requested to post your question in the chat box if you have any. We can take it for our speaker, Professor Dr. Paul. Okay, maybe I can, do we have a question here from the in-person audience? Okay, so Professor Paul, I can see one question from Mr. Salman Ali. He says that, can you trust a PLDDP score of 50 to 70 for small peptide structure prediction from AlphaFold?
So this score from AlphaFold, right? Again, people have different views. I would certainly not trust anything below 70. And we clearly see that rather 90%. So usually the core of the protein that is well predicted is above 90%.
Okay, all right. So there are also very many compliments for your talk. Let me see. Okay, there is a very low. Okay. So there is a question from Mr. Rocco. He's first of all thanking you for your presentation. So he's currently a postdoctoral fellow at ICCBS, which is of course home institute for him and also for you. You are very familiar with ICCBS. So his first question is, how has the integration of AI and machine learning changed the way structural biologists approach target identification and structure-based drug design? This is his first question. Yeah.
Yeah, so we really use, so when people give me a new target as a sequence, we put this first into AlphaFold and Rosetta Fold to get an idea about the protein itself. So for example, we would clearly see if the protein is a membrane protein or if the protein is largely intrinsically disordered. And in both cases, I would say in terms of molecular biology, they're probably too difficult to tackle at the moment. And as I've mentioned, the biggest advantage is that we get a very good idea of what the catalytic domain is for construct design. So which which design or which length of the protein can be used for recombinant overexpression, in particular when we stick to rather simple expression systems such as E. coli. Another thing that I didn't mention, I didn't have time, is of course post-translational modifications. These programs are not very good at predicting post-translational modifications that could be really important for any eukaryotic targets, so NTD targets. And again, the problem is that we simply don't have the database that deep learning methods can use to learn which residues are likely to be post-translationally modified.
Okay, so his second question is, what are the current limitations or challenges in using AI for predicting protein ligand interactions, and how can structural biology help overcome them?
So it's the same thing. So for protein ligand interactions, these methods work very well when you have a large database. So a typical example is kinases. There are hundreds of or thousands of protein structures of kinases. In fact, there's a huge effort at the moment in the United States to use the databases from industry, in particular for cancer targets, again, kinases, to use them for the machine learning algorithms. So basically, all the big companies have thousands of structures of kinases bound to ligands, which are not in the public databases, not in the protein database. So people are trying to use those data sets then to improve the machine learning algorithms for, again, cancer targets. And I think that's going to work. Again, the problem for neglected tropical diseases is that, and I think Paul will tell you more about this, we don't even know exactly what the targets are for some of our therapies. And many of the therapies have probably more than a single target. And at that point, I think that machine learning methods simply cannot work and will not work.
Okay, so the very last question for you, from Mr. Noor Rahman, he's asking, what do you think about machine learning model? If we do not have enough experimental data, will it be reliable or not?
The simple answer is no. And what we need to do is really have more experimental data. I think that's the key point. It comes back that these methods only work if there's a large experimental data set, and in particular, a reliable data set. And I can tell you that on a different projects, we're actually trying to work together with a machine learning expert, because the problem with experimental data often is that different parts of experimental data have different levels of reliability or confidence. And the way of weighting different data is actually quite tricky in terms of statistics. And for the protein database, that's actually quite easy, because we know actually our coordinate error in the protein database. So if we use a protein database, we actually know the coordinate error of every single of our coordinates, or we can at least estimate the coordinate error, which for mathematicians is wonderful. When we think about experimental data in the molecular biology lab, our experimental errors are at best a guess.
Okay, thank you very much, Professor Paul. Thank you very much for your lecture and for taking question and answers. You have another lecture, so we will be waiting for you. But before that, we have lecture of Professor Dr. Paul with us. But now, right now, we are going for a networking tea break for 10 minutes. So all the in-person participants, please join us in the adjacent room for the tea break. For the online participants, you may have it in your labs or in your rooms. We're sorry we cannot provide you virtual tea. But anyways, thank you very much, and we hope to have you here in 10 minutes. Okay, welcome back, everybody. Now we will take the very first lecture of Professor Dr. Paul Denny. Professor Dr. Paul Denny is a professor at University of Durham, UK. He is also fellow of Royal Society of Biology and fellow of Higher Education Academy, UK. The title of his talk is Anti-Protozoal Drug Discovery, Pearls and Pitfalls. So, I request you to please welcome Professor Dr. Paul for his lecture.
Yes. Oh, it's not. Okay, so hello, let's wait for this distinguished host to come in. So yeah, it's a great pleasure to be here, and just say thanks to my good friends, Professor Sam and Professor Iqbal, and comstech indeed for inviting myself and indeed Anchor to speak to you today. Emperor's pleasure to be here, but he's joined us online. And most importantly is thank you to you for coming, either in person or virtually. So very much complementing Emka's talk earlier today. I'm going to be talking about the difficulties of drug discovery in reference to the critical search for antiprotozoals. And I will finish up with some basic information about what we're actually doing in research terms with ICCPS in Karachi. So I'm in the Department of Biosciences at Durham and in the Molecular Interactions Group. And we work very closely with chemistry. That includes Emka and Patrick Steele in particular. So a lot of work I'm going to talk about is with those two individuals. And the factor that brought us together, as Iqbal mentioned, and it's a very valuable thing, unfortunately the funding is no longer with us, is the Grand Challenges Research Fund, which was a British government initiative to bring research collaboration across the world to low and middle income countries. So we have this network of institutions in Latin America, South Asia, and of course the UK. So that really is the basis of all our work going forward in this area. So Emka touched on sphingolipids. Oops, excuse me. There we go. Emka touched on sphingolipids in terms of the enzyme, but just to remind you, does anyone know what sphingolipids are? Or does anyone not know what sphingolipids are? Well, all right, I'll cover it basically. So they're lipids, of course, and they're implicated in many eukaryotic processes, including, this is a kind of classical, very simplified Siemens diagram you can see here, showing a lipid raft, which you've probably heard of, which are arguably only theoretical. The biological evidence is somewhat limited, but they do exist in some form in eukaryotic cells and are very important in a number of roles, including trafficking and signal transduction. So, core essential factors in membranes and function. Now, before I talk about neglected tropical disease and leishmaniasis in particular, I'm just going to interface with what Emka was telling you by talking about toxoplasma. Now, toxoplasma is a very important pathogen of humans and domestic animals like sheep. Big impact in Pakistan, for example, in the lamb and sheep industry. For us, it's a model apicomplexan parasite. Definitive host is the cat. Causes brain deformation, both in the unborn child and indeed in the unborn lamb. And it's related to parasites such as the causes malaria of Plasmodium species. Now, Emka mentioned SPT, or fully it's serine palmityl transferase, and it's the first and rate-limiting step in sphingolipid biosynthesis. It's a transmembrane enzyme, and essentially it does this job here. So it takes palmityl-CoA, coenzyme A, L-serine, condenses them to form ketodihydrosphingosine, KDS, which you can see here. Okay, it's got this amine group there. Now that's important because that is the basis of all sphingolipids. Now, In eukaryotes, there is a prokaryotic version, as Emka briefly mentioned. In eukaryotes, it's dimeric. It has two core subunits known as LCB1 and LCB2. LCB is long chain bases from Saccharomyces cerevisiae, so fairly meaningless names, but a lot of Saccharomyces names are really meaningless, but you stuck with them. So it looks like this. There's transmembrane domains, which you can see here, which hold it in the membrane. LCB1 is non-catalytic, LCB2 is catalytic, but they do not work independently of one another. So they need to be together, and they're both essential for this function. And it's within the inner leaflet of the endoplasmic reticulum. So it's in the lumen of the edam. Now the bacterial serine methyltransferase is completely different. Rather than heterodimeric with two different subunits, it's homodimeric. And soluble as well. I mean, bacteria, of course, do not have an ER, but it's soluble. And the two identical subunits are shown here in yellow and blue. This is a genuine crystal structure, not a model. And again, going back to what Emka was saying, what we've done together, we have modelled the Toxoplasma SBT, and it's within this experimentally derived envelope from small angle X-ray scattering. So we've got the shape of it, and the model fits within. And importantly, it's a bacterial-like enzyme. Of course, Toxoplasma and Plasmodium and all the other AP complexa are eukaryotes, but they seem to have replaced the eukaryotic enzyme with a bacterial version. We can talk about that at lunch if you want. It's a very unique, completely unique part of eukaryotic life. So sphingolipids, I would say the core sphingolipid, which is upstream of serine methyltransferase activity, is ceramide. So you can see the core unit here, the amide, now an amine, sorry, the amine, now an amide, attaching another lipid, which is going out here, C15H31. So it's dually acylated in that way. And this, through this primary hydroxyl group, becomes phosphorylated either in humans and most other complex mammals and related species, becomes sphingomyelin, and we have a phosphocholine attached here. In yeast plants and indeed in some protozoa, we have phosphoinositol. So you see the head group is rather different, that simple choline, and that rather more complex inositol group. Okay. So similar, but significantly different and probably functionally different, but we don't quite understand why. Now I have to show this model here. Now, in Toxoplasma, and indeed in all the other AP complexa we can get sequence data on, we have a complex picture of ceramide synthase. We have one version you can see here, and we have a second version here. So we have two groups, ceramide synthase 1 and ceramide synthase 2. Now, again, getting back to Emka's talk, of course, we can get an alpha fold or Rosetta fold model. This is alpha fold, this is ceramide synthase 1, this is ceramide synthase 2. In magenta, these are the active site residues. Now, but what we know from the sequence is that ceramide synthase 2 has changed these active site residues, which would, we think, render it non-functional. But the fact we can say that from the sequence and model it so they're in the same place as the functional enzyme, but it's not good enough. We have to actually express these in vitro and do a functional assay to prove that was the effect of this change. And this is leading on to other work. And also, if we really want to understand what happens in pathology, we have to start using genetic engineering to remove these enzymes from the parasite and produce a line where they're knocked out. And as we can see here at the bottom, the ceramide synthase 1 knockout in the control, because of the engineering, which I will go into. Before knockout, they're red, and when we induce knockout, they become green. And similarly, ceramide synthase 2 before red and after green by fluorescence. Now, the important image here really is this one. These are little 96 well plates. You can see the plaques form when infected with toxoplasma. This is after 72 hours, I think. Rapamycin induces this effect, induces the knockout. We can talk about why later on if you wish. And you see the knockout of the functional ceramide synthase 1, the one that actually works as a ceramide synthase. It doesn't do very much. It's very similar to the control parental line. But if we lose ceramide synthase 2, we get rid of almost all pathogenic, self-disrupting activity of toxoplasma. So whilst it's not functional, it's very important. And surprisingly, whilst functional, ceramide synthase 1 doesn't seem to be very important at all. So we published this and we're now sort of looking to take this forward. And actually, Emka and I, we're going to submit a grant on this literally when we get home. Okay, but I'm going to now focus on leishmaniasis because that's been the main focus of my work for a number of years, and particularly a focus of work with ICCBS and the broader global network. So important to know, there's a very sharp uptick in leishmaniasis, particularly cutaneous disease. The number of countries affected is increasing. Why, you may ask? Well, it's a vector-borne disease. There is a movement of the vector due to deforestation, due to climate change. There's a movement of people. We saw that most recently in the Syrian conflict and the diaspora, with cutaneous disease becoming quite common and being transmitted in Lebanon, for example. The number of people at risk, I mean, this is put anywhere between 350 million and 1 billion, including many people in Pakistan. And it remains a critically neglected disease. And this is something I saw when I was in India in 2018-19. This is a teenage boy with visceral disease, Kala Azar, black fever in India. Still there in Bihar in northeastern India. So it remains a problem. You will hear that the disease has gone away. We don't quite believe that at this point, but we will see. Okay, so this is my own personal sort of contact, if you like, with the problem. This was back in 2019-20, on my first visit to Karachi. We were lucky enough to get a trip to the Jinnah Medical Centre, particularly the dermatology ward. And these 4 children, whose ages were between about 3 and about 8, had returned to Karachi from a rural trip to see grandparents, and they'd all had lesions. And you can see the different lesions here. This one's healing, but scarring. This one's about to heal. This one's healing quite well. This one's not healing at all. So we can see the different responses, and we can see the way this affects the facial region of these small children, but it could be an adult. And these kids were kept off school. Their father, a day wage labourer, was kept off work, so he had to look after them. For those of you that are not familiar with leishmaniasis, as I mentioned, it's a vector-borne disease. We have flagellated promastigotes biting. Could be a human, could be in most species another animal, such as rodents or dogs. They form animal reservoirs. And they invade macrophages, where they lose the flagellum, or at least an obvious flagellum, and they become amastigotes. And this is the form, be it in the liver and spleen, or in skin or mucosal membranes, that causes the disease: visceral disease, cutaneous disease, mucocutaneous disease. The type of disease depends on the species of the parasite, the site of the bite, but also the immune status of the host as well, and their genetics. Some quite profound differences across the world in the effect of these. Now, these are the drugs, and AI is not going to help rescue any of these drugs, I'm afraid. You'll see at the top, this is what you will get in the Jinnah hospital. If you go in, you will get injected with these antimonials, very toxic, been around since the 1940s, so getting on for 100 years. In India, for visceral disease, they're using a one-shot liposomal amphotericin B. See, this is quite a complex. This is a natural product. It was identified as an antifungal. It's quite a specific mode of action. The only one of these that does, and it is appearing to have some effect in India and Nepal and Bangladesh, but we don't know at the moment whether that's going to be long-lasting. Paramomycin, still in use, an antibiotic, a natural product. You can see here, now used in combination, particularly with miltefosine. Miltefosine is the only one of these that is non-injectable. It's the only oral therapy. We're particularly interested in miltefosine in our lab. Pentamidine is actually fallen out for general use because it's too toxic. Okay, so this is our interest in miltefosine because it's very closely connected with particularly sphingolipid and sterol biosynthesis. Okay, so let's focus on the protozoan inositol phosphoryl ceramide with us head group here, because that is the difference. So there's similarity up here, and then when we get towards ceramide in humans and other animals, we have sphingomyelin produced by sphingomyelin synthase with that phosphocholine group. And yeast plants and some protozoa, including Leishmania, we have inositol phosphoryl ceramide, as I said, produced by IPC synthase. And that essentially is taking the phosphoinositol from phosphatidylinositol, the phospholipid, and putting it on the primary hydroxyl of ceramide. Now, in Leishmania and in other eukaryotes, sphingolipid biosynthesis is very polarized. So this is labeled as ceramide, the substrate, or one of the substrates of IPC synthase. And you can see the concentration in the Golgi apparatus. This is known, for those of you not familiar with kinetoplasts, as the flagellar pocket. And this is where all the traffic, the material, the lipids, proteins, leave the cell. And when anything, food or whatever, comes in. This is its only door out and its only door in. And you can see that sphingolipid biosynthesis occurs very close to this, because the complex sphingolipid is going to be arrayed around the plasma membrane, which includes the flagellum in the case of the promastogome. Now, if you're thinking about what the enzyme does, I already mentioned some of this. There's an important balance factor here. So this is a very diagrammatic representation of the enzyme. So this is the enzyme non-functional. It doesn't exist like this in the cell for very long, and it's very intermittent. So it has an activated histidine here, okay, in its active site, which is fairly well defined. We have no crystal structure because it's a complex multi-membrane protein, and you'll hear this term quite a lot today. There's 6 transmembrane domains. It's in the Golgi apparatus, as I indicated. But what we have is the phosphatidylinositol, this phospholipid, duly acylated, coming in, phosphoinositol. It captures the phosphoinositol on that activated histidine, releasing diacylglycerol, or DAG. Now, at least in mammalian cells, we don't quite understand this in Leishmania. DAG is mitogenic, it promotes cell growth. Now, on the other side of this equation, we have this activated histidine with the phosphoinositol. Then we have ceramide, remember that primary hydroxyl group. It comes in, it captures that phosphoinositol, releasing the enzyme to go back again and producing the product IPC. And ceramide, in contrast to DAG, is cytotoxic or apoptotic, at least in mammalian cells and indeed in plants as well. So we have growth promoting production and cytotoxic apoptotic reduction in ceramide. So we have a balance between life and death at the cellular level. So that suggests it's a good drug target, and that's what we based it on. And since the 1980s, really, people have been working on this in fungi. A number of natural products identified to inhibit the fungal IPC synthase. known as all-1 in yeast and other fungi. Probably the primary, and the reason it's called all-1 is this complex deoxypeptide here, oleobacidin A, which you can see is definitely a natural product. That's gone close to clinical trials, but has never quite made it, although I think it may have a new life in the not too distant future. Okay, so we had a complex membrane protein. We wanted to consider it as a drug target. Now, broadly, we can think of drug screening in two different ways. We can think of classically in a pharmaceutical industry, it would be in vitro high throughput screening. So that is a classical biochemical assay in a multi-well plate, screened with thousands or maybe hundreds of thousands of compounds. Now, the advantages are it can be very simple, specific. You're looking at enzyme activity, looking for chemicals that inhibit that activity. And also have knowledge, so you know the protein is interacting with the ligand, the chemical, and we can facilitate structure-activity-based relationships quite easily. The disadvantage, it can be really difficult, such as a transmembrane protein, to isolate the protein at levels high enough and cheaply enough to actually do this, and it can be very expensive, and you have to have specific substrates and read them. So very common, and what many people do, particularly in the world of neglected disease and phenotypic HTS, so literally throwing your compounds against cells, be they leishmania, cancer cells, bacteria, viruses, whatever. So you don't need a purified target. You're looking at everything in a cellular context, and you're essentially screening all targets at once. And as Emka said, most of the drugs we have hit more than one target, at least for leishmaniasis. But they can be quite insensitive because cells tend to reject these foreign chemicals, and we don't have a target, so there's no structure activity basis, so we have to think about the target later on. Now, in our work, we looked at yeast-based HTS, so we express the protein in yeast and then have a readout, so we can look at the functionality. So this combines some benefits of the in vitro system with the benefits of HTS. You don't need a purified target, but it does give a cellular context, a eukaryotic cellular context, and also yeast, cheap, easy to grow, straightforward to manipulate, and we can discriminate fairly easily with some genetic tools, false positive hits, but they can be insensitive due to drug pumps and their thick cell wall and other barriers, and we have to have our protein functioning in the cell. So it's no good having expressing a protein in the Saccharomyces cerevisiae, and it doesn't work because it's in this non-native cellular environment. So remember the Leishmania IPC synthase, which I hope I've convinced you seems like a great drug target. Well, because of this transmembrane nature, we couldn't do sort of biochemical HTS, high throughput screening, so we went to a yeast system. So in that we started with a deletion of the all-1 supported, that's the yeast IPC synthase, supported by expression of all-1 on a plasmid. This has a URA3 marker, which means it can be selected for in the absence of uracil in quite easily on literally an agar plate. Then we add in the Leishmania IPC synthase under a galactose promoter, so it will be suppressed in the presence of glucose, activated in the presence of galactose, with a different marker, LUM2, to grow in the absence of leucine. So we get both of these present in the cell. And then we can do something called a plasmid shuffle. Again, this is fairly easy. So we have something called 5-fluorotic acid. And actually, when the URA3 marker its biochemical activity interacts with 5-FOA, it produces a toxic product. So you purge the yeast of this, and then by growing in galactose, you have yeast that are fully supported by the Leishmania enzyme. So this worked actually perfectly. And again, thinking of a control, because we have to have a control, we also engineered the equivalent strain with, rather than the Leishmania IPCS, we have the yeast or one here. So we've got a control. It'll become clear wine very shortly. So having that in our hands actually allowed us to start working with GSK in Spain, as it happened. At the time, that was where their European screening centre was. I'm afraid it's closed and moved to Philadelphia now, which is disappointing, but there we go. So we started the whole library, which is about 2 million compounds. So this is quite, at the time, for me, almost unbelievable. There'd be a library of 2 million compounds and we could screen them against an enzyme. But that's what we did with funding from their Open Lab Foundation. And then we screened them against the yeast with the Leishmania enzyme, and then we screened them, the hits at least, against the yeast with the Leishmania, and as a counter screen, the yeast supported by the All-1, the control I mentioned in the previous slide. So then we go quite quickly from 2 million down to about 4,000 compounds, and then we do a dose response. These are all at single concentrations because of cost and time, and indeed material. And then we have a readout at that point before we go to dose response of either non-active, because we don't want those, active against All-1 but not IPC synthase from Leishmania. They're really, really interesting, and we would like them, but they didn't give them to us because these are potential antifungals. Active against both, this is the critical one, because if they're active against both, chances are it's just a generic toxic antifungal, and they will give us no data at all, really. And then we want ones to prioritize those that are active against the yeast supported by the Leishmania enzyme, but not All-1. These are the ones that are going to be specific for our target, the Leishmania IPC synthase. Okay, and it's only then that we go to the selectivity ratio with about the 4,000 compounds we have. And it's shown here on this diagram. So all of these figures are in log values, okay? So 6 is 1 micromolar, 7 is 100 nanomolar, 5 is 10 micromolar. So essentially this is plotting the EC50 concentration that kills 50% of the yeast against the All-1 yeast up here and against the Leishmania yeast along here. Now, if they're both the same, they'll be on this diagonal. And I already told you we've selected those that are preferentially hitting the Leishmania complemented yeast. So they're all to the right of here, and particularly want the most selective. These are our most likely candidates. And the selectivity ratio, again, log values. So this is about a selectivity of 35 up to about 300. Okay, all in this bottom right-hand corner here. So we're down to 500 compounds. These are our hits. Just to give you an idea of what happens at industrial scale with these things, we go down even further. We do something called clustering, which is where we take a database view of the compounds we're looking at, and we look at those that are very structurally similar, because what happens in industry and indeed in academia, small changes are made to a target molecule, like target compound, and you'll get a whole series with just a hydrogen lost here, an oxygen here, etc. So try to reduce those and remove them, so we end up with 2 compounds per cluster, and plus all the ones that are just singletons out on their own. That leaves us 211. And it's only then that we go into the rather more laborious process of screening against parasites. And to make this easier, remember I said that amastigotes are the pathogenic part of the Leishmania life cycle, and they invade macrophages, but we use azenic amastigotes so they can grow free. They're not proper amastigotes, but they grow free in culture, and we can get a much readier, easier outcome. And again, log criteria. This is a log value. Again, it's the EC50, the concentration that kills 50% of the leishmania. This means it's less than 10 or equal to 10 micromolar. Okay, you'll see that 10 micromolar figure again and again, particularly in an industrial context. That's the start point. The property forecast index here. This is kind of mystery. I'm not sure what the equation is because it's a GSK secret, but this is what they use based on size, hydrophobicity, kind of like Lipinski's more sophisticated rule of many. And they have a value that's in log terms that is less than or equal to 8. The number of aromatic rings, very important, driver of hydrophobicity, less than or equal to 4, and critical is this again, a log value selectivity index, which means here they have to be selective tenfold over these HepG2 liver cell cultures. Okay, so all of these we go from 211 to 25. Now it's only then we actually see something that looks like reality, to be honest, and then we go into a very sophisticated, because we're working with GSK, we can do this intra-macrophage screen. So we look at the small number of hits, the 21 we have at this point. We screen them against infected macrophages. So the green is the amastigotes. The purple is the nucleus of the macrophage, and you can just see the outside, the limiting border of the macrophage shown here rightly. So with a smart camera and computer system, you can count the number of macrophages infective and the number of the amastigotes within each macrophage. Okay, so we get a lot of data from here. Again, we're looking for an activity of less than 10 micromolar and selectivity, this time against the macrophages themselves, of more than 10. So then we get 5 hits. So we've gone fairly quickly, actually, from 2 million to 5 in this project. 2 series, including these benzazopanes, which you can show here, which we were taking forward, and 1 singleton. Now, the yeast, it might look really attractive, but it's still an indirect assay. You're assaying a protein expressed within a foreign cell. So what we do, which is not often done, with a few people that are doing these kind of assays, there are some exceptions, is then use a biochemical assay, a very complex one, as it happens, not suitable for high throughput screening, and which involves the use of a fluorescent ceramide, MBDC6 ceramide, in the assay, semi-purified enzyme we produce, labeled IPC. And for this, we showed that the bezazopanes and some of their derivatives from the broader GSK library all had sub-micromolar IC50s, inhibited enzyme concentration, enzyme activity by 50% or at low micromole, sub-micromolar levels for the benzazopanes. So we went all the way from the assay all the way through to the benzazopanes identification and then proof of activity in a vitro assay. So that looked really good. So we actually were invited then to go into what was called the Wellcome Trust Drug Discovery Engine to move early stage research, still early stage, to the next level. Now, very difficult. We had to actually get close to matching miltefosine as an oral drug for leishmaniasis in an animal model, mouse. And at that point we failed, so we didn't get to the required 70% efficacy compared with miltefosine. We got to around 30%. So we didn't fail in the project, but one of the pitfalls of these kind of projects is you're very likely to fail. This is why it costs billions to make drugs, and that cost, because of AI and other initiatives, will come down. We've seen that with vaccine development. But it's still a big barrier, actually, to taking things forward. So we published it, and also we worked through the network with our colleague Silvia Uliana in Brazil, in USP, And tamoxifen, which you may have heard of as an anti-breast cancer agent, actually does inhibit the same enzyme. And working with Patrick this time, and a long-standing project with a company, well, then MRC Technology, now LifeArc, small library, which we did screen. We identified clemastine, which is an antihistamine. And yeah, we identified it as an enzyme, an IPC synthase inhibitor. Also, it showed similar inhibition of lesion development in a mouse model. This is Leishmania amazonensis, and reduced that similarly to glucantine, the sort of benchmark, which is one of these antinomials. Just to introduce, though, we are interested in mode of action and working with Mike Barrett in Glasgow in particular. We look at, through metabolomics, changes in specific lipids or other small molecules. And we see an increase, for example, in ceramide. Remember that ceramide is one of the substrates of the enzyme. And what we can see is a very significant increase in ceramide, which is exactly what you'd expect if you inhibited one of the enzymes which uses that as a substrate. Okay, and then we got the front cover. Now, but for all this, I haven't actually told you that IPC synthases are a drug target in any genetic way. In fact, well, work from me when I was a postdoc, well, over 20 years ago, knocked out the SPT2, as we called it, which is one of the subunits of the Leishmania SPT. Gets rid of activity and indeed IPC synthase. And what you can see is, well, you know, this is the knockout. Yeah, they do sort of slow to actually produce lesions, but they do eventually, and they actually match pretty much the parental and the outback controls. And similar results from Steve Beverley's lab around the same time or a little bit later. So is it really a knockout? I could tell you lots of why I think it is still a target, but this is probably the most interesting and just shows a diversity of approaches. So we took the knockout that I'd created over 20 years before for the Leishmania LCB2, that's SPT subunit, and we sequenced the genome because I suspected we had non-targeted deletions because we were understanding at this point that the Leishmania genome is very plastic. So this is actual genome sequencing. So you can see the depth of sequencing, and also you can see in the knockout this gap here, completely missing. Some misalignments here shown in red arrows, but this is completely missing. So as well as the LCB2 SPT subunit, we have this missing. And this is a putative ABC transporter, and putatively ABCA3, which in humans is a steroid transporter. So to provide some validation for the fact that this, which was in all the knockout lines, was vital for the loss of the LCB2, we tried to use modern technology, gene editing, CRISPR-Cas9, which I'm sure you've all heard of, in Leishmania mexicana rather than Leishmania major this time. But we couldn't knock it out. It was impossible, multiple attempts. But when we actually knocked out this ABCA3 ortholog together with the LCB2 at the same time, not separately, at the same time, we were able to knock out both. So our hypothesis is this is compensatory. Now, what about the IPC synthase? Well, again, Steve Beverley's lab, a long-term nemesis of mine, hopefully he'll retire soon, is the inositol phosphorylceramide null, Leishmania are viable and virulent, which he shows there. In fact, he shows them to be hypervirulent in mice. Now, this is actually knockout lines generated not with gene editing and CRISPR-Cas9, but old school homologous recombination and drug selection, which takes months, essentially. And also, we have shown leads to unexpected deletions in the genome, such as the LCB2 knockout I showed you earlier. So around the same time, published rather afterwards, we evaluated this in Leishmania mexicana using the gene editing CRISPR-Cas approach. What we see, yeah, we get lesions in the parental line, which is this one here, and we get a loss of lesion growth down to 0 in the knockout, and unfortunately the add-back line, the reconstitute activity, also was 0. So a complex picture. We have some ideas why this wasn't, the add-back didn't go back to parental. But anyway, this indicates that yes, the IPC synthase is essential, and maybe we should think about using the gene editing rapid knockout production technology where we don't give the parasite the chance, such as in the conventional system with the molecular recombination and months of work under drug selection to change their genome. Genome plasticity is something we have to be aware of. And as part of the network, both of these projects were part of a network project with Santuza Teixeira in Belo Horizonte, Brazil, looking at related parasite, Trypanosoma cruzi, the cause of Chagas disease in Latin America. CRISPR-Cas9, again, we get a very similar outcome. So the wild type produces parasites which are in the serum, knockout gone, add back doesn't recover. And similar here with the survival of the mice. So both the knockout and add back survive. The parasites are not viable. And even though we can see that the add back does recover parasites in this assay in heart tissue from the add back, the knockout situation, this is not replicated in vivo. And there's a complexity of lipids which we can talk about later. But suffice to say, there seems to be some hardwired reorganization of metabolism, some of which we've picked up, although it would be very complicated to go further than that. Okay. So back to this, I'm going to finish up the drug thing with really thinking about the failure of in vitro techniques and our use of yeast-based HTS, which I described, and described that as a failure, although the project worked, and then go back to phenotypic hits, because the general experience, this is 10 years up to 2008, was these are new medical entities, okay? So these are drugs that went into the pipeline. So of target specific, there's new stuff, about 60%. Substrate analogs, that's just taking known substrates of an enzyme and just making small analogs that will bind to the enzyme and inhibit activity, maybe 10-15%. And then the third remaining is from the phenotypic assays. But when we think about first in class, these are the really new chemicals. These are not things that are slowly related to other chemical entities in the drug pipeline. We see that the situation between phenotypic and target base is reversed, with 60+% of these first in class being from the phenotypic studies. So this drove our friends at GSK, Tres Cantos, to actually take a phenotypic approach to the 3 kinetoplastid parasites. And also, again, natural product interest. GSK will not touch natural products. I've had a conversation with them, but you will all be aware, I hope, in 2015, the Nobel Prize was shared between Campbell, Amura, and Tu for the discoveries, respectively, of avermectin, which becomes ivermectin for nematode worms, and probably most critically, that's why you two got half of this, artemisinin for malaria. It's still the core anti-malarial for much of the world. So I already mentioned that amphotericin B and paramycin are natural products themselves, core anti-leishmanials. So just to finish up, before I just talk very briefly about diagnostics, we have worked in the natural product area with this company, Hyper Discovery, for one. These are fungal secondary metabolites, low molecular weight. And we do a phenotypic approach. We screen them against these A-xenocaine mastigates, for this time from the Lithuania mexicana, and some mammalian cells for cytotoxicity. The same approach we use for the GSK. We sort of took a lot of learning from that GSK project. After lots of separation, HPLC, rescreening, we ended up with one compound here. So it maintained activity in the macrophage model. That showed the black line over the red line. And it was marginally better, largely less toxic than against uninfected macrophages. But this index of 5.5 is not good enough, really, to take forward for any further study. So we did do, for the sake of the project, some mass spec and NMR structure elucidation. So it's a novel natural product, bisabolane, sequetapine, a lot of oxygenation and saturation and unsaturation in it. So, and we did do some polyomics. So we looked at the metabolomics I mentioned. Again, we did this with Mike at Glasgow. And what did we find? Well, the parasites of the kinetoplastids, such as Leishmania, have this trypanothione peroxidase system. That's how it deals with the oxidative stress. It's their integral redox system. Central to this is trypanothione, which you can see here, which is the equivalent of glutathione in mammalian other systems. So trypanothione was almost absent in the on the treatment with our identified compound, as was its precursor glutathione. It was over 60% down. And also, there was an oxidized cysteine derivative, cysteine, identified. This is all indicative of oxidative stress, although we have limited data, to be absolutely honest. However, if we look again at the structure, there's these two alpha beta unsaturated ketones, which you can see here, that indicates they're Michael acceptors, possibly reactive towards nucleophiles such as trypanothione and glutathione. So our hypothesis was they're actually taking that out of the system and leading to this oxidative stress. So I just want to finish up just talking very briefly about an ongoing project between myself and Samir Yusuf at CBS. So one of the problems we have is antimonial therapy. This is being injected directly into this child's lesion, you can see here. But the antimonials against Leishmania tropica are up to 50% really ineffective, leading to the kind of lesions that we saw in those children, where some resolve and some do not, even after treatment. We do have a mix of tropica and major Leishmania. So major is fairly easy. I say easy, it's true. But tropica difficult, also a chronic disease. So what we are doing is we're developing a molecular differential assay so we can identify people infected with major or tropica, and we can start to think about more specific treatment. Diagnostics are very important, particularly probably in neglected tropical diseases, to ensure that the rather poor treatments are used more effectively. So we decided on the basis of evidence and push and the need for something simple and cheap to use loop-mediated isothermal amplification of LAMP, old PCR essentially. It does work. We have sets of primers which we've shared with Sama for their project, which parallels ours, which differentiate between major and tropica. And as well as working with the ICCBS, we're also working with Marlowe's Peters group in Manchester now, on this to try and get a cheap device. And we've also connected with a company, Video, who actually have a device, so we're evaluating that. So we're moving in that direction. And just to say, very important, we're part of a broad team. And last year, great that Samer and Iqbal were both there. We won the Outstanding Team Impact Prize in the MRC for our NTD network activity. So thanks to everybody and the funders, and any questions, glad to answer.
Thank you very much, Professor Dr. Denny, for a comprehensive presentation. The floor is open for question and answers. For the in-person audience, we'll take your questions first, and then we will move to the participants who are here online with us. You are requested to please post your question in the chat box. So do we have a question here in this audience? So kindly introduce yourself before your question.
Assalamualaikum. Good afternoon, everybody. I'm Dr. Hassan Arouj, a former Director General of Health Services. Presently, I am Post Director at Health Services Academy, the premier public health university in Islamabad in Pakistan, and also an external faculty of Baylor University Public Health Program. So thank you very much for a very insightful presentation. With your rich experience in Pakistan, Jinnah Hospital, and India, do you have some sense of idiosyncrasies which the patients have developed during your experimentation or during your research with the same medicines, and have they been compared with other regions of the world for any change in the dose or their mode of action based on the efficacy of the drugs? One. Second, very shortly, there's a lot of clinical research which is also being done in developing countries, but I don't see a real, maybe, you may also contribute to responding that I couldn't see a real outcome of that. A huge amount of money and, millions of rupees and dollars are put in. So has some organization tried to explore the root cause of this failure? Thank you.
Well, I suppose, firstly, I mean, we're at early stages with, although I met those patients, 6 years ago now, we're at early stages with the clinical work. Professor Samuel and I were talking about this, I think, last night. Saturday night, and we don't have the data to really understand why. Now, I think a good example is Leishmania donovani, and there's a number of pieces of information here. Okay, so firstly, Leishmania donovani causes the India, Bangladesh, and Nepal to restore leishmaniasis cluster, which is being treated with liposomal serine B with some success. As I said, we don't really know how much. Now, so that's regarded as having worked with in both India and Bangladesh having declared themselves not eradication, but not eradication, not elimination, or the other way around. I can't remember. But anyway, that's been regarded as a success. But in Sri Lanka, the same parasite is causing spontaneous disease. In East Africa, particularly Ethiopia, where most of the work is ongoing, Serine B is not working in the same way, so it's not clearing infection effectively. Now, we've got very different genetics between East Africa and Bangladesh, India, and I don't know what the difference in genetics is between Sri Lanka and those regions, but presumably that's rather due to their genetics and immune response than any other factor. We hope, with numbers, that we'll get some understanding in Pakistan as to what's going on, particularly with the impact of different species, Tropica and nature, and possibly a mix of infection. We think at the moment that the Tropica is becoming more prevalent from limited data, and that is causing an increased treatment failures. But we'll find out. And there's not many people working in this area. Actually, it seems that which gets me onto which maybe Iqbal has a more global view than me. Yeah, I mean, the failure, the amount of money that's going into what are very complex diseases with sociological as well as medical problems is limited compared with, you know, cancer treatment, diabetes treatment. And I think one of the mistakes has been to believe that drugs are the only solution. There'll be limited vaccine investment. That may change, although funding for all these diseases has diminished in the last 5 years, the wave of COVID, if you like. But also the investment in diagnostics was pitiful, which is why we want to do what we're doing at the moment, the diagnostic development. Thank you.
Okay.
And, Dr. Rooj, the effect is that the kind of investment required to develop a new molecular entity is really huge. We're talking about literally $3 billion USD for development of one single so the cost of failures, which Paul has mentioned. I don't think any developing countries would be able to afford that. So as far as the research is concerned, it is fine, but when it comes to the development stages, there is a gross failure on the funding. Because that is the amount of money which is required to develop something to a clinical level is not there set. So, and even countries like India and China, you don't really see many drugs coming from them as well. The drugs are largely coming from these large pharmaceutical conglomerates. These huge investment, because for them it's just a business, which of course, as Paul mentioned, focusing more on prevailing diseases and not India. My question to you, Paul, is that you have actually mentioned very nicely that the difference between target specific and phenotypic and specific approaches is better. There is kind of an obsession and also compulsion. The pharmaceutical companies are obsessed on target specific. While in developing world, very large number of scientists would perhaps work on phenotypic because this is the easiest thing which we can do and afford. Can we really marry two of them together? Can we use phenotypic screening for identifying the compound and to eventually identify the targets? Can we do that?
I think it's becoming, whilst still difficult, it's becoming easier, and I think pharmaceutical industry is responding slowly to some of the failures of target-based screening and some of the possibilities of phenotypic screening. In a general sense, we're not talking about neglected tropical diseases. So the main point is the development of technologies, genomic sequencing to look at mutations that lead to resistance. And then such very, very new things, thermal proteomic analysis, where we look at literally the binding of a ligand, a candidate compound for development against the entire proteome by mass spectrometry. We've also done that. That's expensive and difficult, but it's becoming cheaper and more routine as it develops. So there are tools that are coming online which probably will encourage more use of phenotypic screening. But there will be a very critical analysis. The natural product I showed you, it's going to be almost impossible for that to ever be a drug. In fact, I think it's impossible for it to ever be a drug because of its hyperreactivity with all sorts of things, and hence its very narrow selective index.
Sure.
So just one last question.
Good afternoon, everyone. I'm Shreddy Salim, PhD scholar from Kajatam University and research scientist from National Institute of Genomics and Cross Biotechnology University. My question is related to the maestro. How long does this trial program for testing this drug? And second one, that whether this drug has some side effects like on mice, like behavior changes or anything observed like that.
Yes. Yeah. Yeah, it doesn't clear infection. We haven't really done the longitudinal analysis of whether the parasites reactivate. Yeah, clemastine itself, the antihistamine, makes the animals sleepy, as you'd expect. But we're now, this is where Patrick's expertise comes in, got a derivative that doesn't appear to make them sleepy. So, I think we've got some data, he'll know better than me, that shows it's not actually interacting with the H2 receptor, I think is the one. So, yeah, important question actually, because repurposing looks great, but you've got to be aware of side effects, which particularly when you're upping the dose.
Thank you very much. Now we move on to the next and the last talk of this workshop. The title of the talk is New Enzymes and Structures from Virusphere, and this will be delivered by Professor Dr. M.K. Paul. You're already familiar with him. He's a co-director and a Durham lead for EPSRC at University of Durham. Professor Dr. Paul, over to you.
Okay. Hello again, and thank you, Paul, for a brilliant talk. Just to check, can you see my presentation and hear me?
Yes, absolutely.
Good. All right. But thank you for giving me another opportunity for a slightly different take on structural biology. But bear with me. At the very end, I will come back to what Paul has said and back to neglected tropical diseases. So this is a slightly different project that we have been working on for a number of years within a European network, and it's called Structural Biology of the Virus Sphere. And the reason we looked at the virus sphere is that, as you all know, viruses are subcellular organisms, parasitic intracellular life cycle, but they require the cellular machinery of its host to replicate. Now, viruses are really highly successful. They do infect all other forms of life. And at this point, I actually have to ask you, Paul, are there any viruses that infect your parasites? And you might want to answer that at the end of the talk. So you've got about 25 minutes to think about this. They do replicate and mutate very quickly. And in fact, they are the most successful life form on the planet. Numbers of viruses are estimated to be more than 10 to the power of 31. And that means that there are far more viruses on the planet than stars in the universe. Now, this is interesting for us because they do represent the largest reservoir of genetic material. And I've spoken about the genetic material already in my previous talks. So basically here we have a vast reservoir of genes that have been involved, and most of these are, of course, not sequenced and unknown. And the goal of this overall project was to really find some of these genetic material and see what we can learn from that. So within this project, the question is, where do we start? And because I had a number of friends in Iceland, we actually started in Iceland, up here in this little village called Hveragerði, which is basically where the two tectonic plates move apart from each other. And you probably have all seen the latest volcanic eruptions up there. The reservoir that we were looking at is on this one up here. This is actually the European team in the vicinity of Hveragerði and one of these hot lakes. Now, these lakes are close to boiling temperature and usually very acidic, high salt concentration. So basically, we were looking into these lakes, and you can actually see some of the members with like 5-liter canisters taking out samples, which were brought into the lab for metagenomic sequencing. So the idea is that rather than looking for a particular enzyme, the entire genetic material in this lake is being collected. We filter the viruses out and then sequence the viral DNA in order to find new enzymes that have evolved for new functions, right? The other place that we looked at with our friends in the University of Bergen is called Loki's Castle. Loki, of course, well known from several Marvel's movies. This is a field of black smokers in the Atlantic Oceans. They were only discovered at the beginning of this millennium. They're located more than 2 kilometers below the sea level and have temperatures up to 300 degrees Celsius and pressures of more than 200 atmospheres. So very extreme conditions. And what the team in Bergen does, they do have these remotely operated vehicles that they send down into these Loki's castle. They leave samples, they take samples out, and then bring them up to the surface for metagenomic sequencing. And I should mention this is done by Professors Ida Helen Steen and Ruth Sander from the University of Bergen. So on the next picture, I actually have a picture a video that they sent us. These are several tens meters high, and you can see these really hot water coming out. And these little flecks are actually critters. So it is absolutely fascinating and surprising how many life forms live around these lakes. I'm going to play this one more time, if I can. There we go. And the interesting thing for us is that within several tens of meters, the conditions already change drastically. So whereas we are here at 300 degrees, basically at the top, we just go a couple of meters away, and then the temperature rapidly drops to about 0 degrees. And also the oxygen concentration changes and so on and so on. So within this microenvironment, you already have a large number of different habitats. So placing then your samples at different distances to these vents, people can then, or Ida was able to collect quite different samples. So basically, we're coming out with a database of several hundred or thousands or millions of genes. And in fact, this was done by the University of Bielefeld, our partners, called into a VirusX database. And right now, at this point, the VirusX database has about 50 million open reading frames or potential genes that need to be exploited. So from that point of view, of course, the question is, how do we actually find the ones that are interesting? And what we did is we looked at two different classes. One were novel gene products with no detectable sequence identity. And the aim was really to discover new functions, new applications, and new biological or molecular mechanisms. And I can tell you when we started, we certainly hoped to discover the next CRISPR-Cas system and become extremely rich and famous. I should tell you that we didn't. We did, however, discover a CRISPR-Cas system from hyperthermophilic organisms, which is very similar and which is also being exploited by our partners. But at the same time, we also looked at proteins with known or putative known biological activities with particular desirable properties. So this was really led by our company partners, for example, Arctic Slime, SME up in Norway, and they looked at lytic enzymes, ligases, recombinases for biotechnological applications. And mark this for a moment because I'm going to come back to recombinases and how we are hoping to use recombinases for biotech applications at the very end of our talk, right? So our job together with Lund and Stuttgart was the production, biophysical and biochemical, and obviously in my case also the structural characterization. And I'm going to give you a couple of examples of what I would call weird and wonderful enzymes that we discovered and hint to some of the applications. So the first one we looked at was actually from a Bacillus subtilis prophage, so not an extremophile, which is a protein called ZAP-A, which has been known to be part of an escape machinery for bacteriophages for quite a long time. But based on the sequence, we couldn't figure out, or nobody could figure out what the mechanisms is, and it really didn't look like any of the lytic enzymes that we had seen before. Now, the gene was shown to be essential for sialysase, but then when we made the protein, the biochemical characterization really showed no detectable catalytic activity. So this is an example where I think the crystal structure was really necessary to at least have a hint of the biological function. I should say we solved this a couple of years ago. So for the experts, we determined it by classical heavy atom phasing, which was quite cumbersome and took a lot of time. Today, we might have been able to use AlphaFold, but I'll come to that in just a moment. So this is the structure, the crystal structure of ZAP-A, and it has basically two domains that are linked by a large linker molecule. So these two domains are both beta sandwiches, well-known domains. And of course, at this point, the first question is, what would AlphaFold have done with that? Well, obviously, we put this into AlphaFold, and AlphaFold clearly, or Rosetta Fold, clearly predicts these beta sandwiches as beta sandwiches. But of course, where AlphaFold and Rosetta Fold fail is the linker region, and also a lot of the loops are not quite correct. So the question, could we have used AlphaFold as a starting model for a structure solution? Well, possibly, but maybe not. Because what happens with this protein actually forms a wonderful pentameric structure shown here. So basically, we have a homopentamer where this linker region really forms a bit of a funnel between these two domains, upper and lower domains. And AlphaFold by itself would not have been able to predict that this is a pentamer. Now, I should say, if we tell AlphaFold that we expect to be a pentamer, the AlphaFold model actually looks quite similar. And we were actually quite impressed by that. So would AlphaFold have been able to solve the structure if we had tried a monomer, dimer, trimer, and so on? We probably would have solved it a bit quicker. So AlphaFold would have been a really nice tool in terms of solving our crystal structure. Now, the other thing that we already mentioned is that AlphaFold cannot give us any sense of dynamics. And because we worked on this for quite some time, we had multiple crystal structures, and these overlaying these different crystal structures gives us actually an idea about how the domains move with respect to each other. Now, these domain movements look relatively small because it's a relatively large protein, but in fact, these loops move by several angstrom, maybe 3 or 4 angstrom. So basically, we can see that the upper domain moves with respect to the lower domain or vice versa. So multiple crystal structures do give us a sense of dynamics, which a single AlphaFold model would not give us. Now, is this relevant for a function? Well, we'll see maybe later, a bit later in this talk. So how do we come from structure to function? And the first thing that we can actually look at is electrostatic surfaces. Because when I'm just going back up here, when we show this structure to non-structural biologists and ask them what the function of this protein will be, the most common answer I get is this is a membrane protein. So people look at this and think, oh, this looks like a transmembrane region with this one part of the domain sticking out of the membrane here and here. But if we look at the electrostatic potential, we see that in particular this part is highly charged with negative charges down here and positive charges down here. So clearly from our electrostatic potential, this cannot be a membrane protein. This has to be clearly a soluble protein. And I should say also in our biochemical assays, it doesn't really behave as a membrane protein whatsoever. So what else can it be? Well, if we look at from the top up here, we see that a lot of these parts are highly positively charged. In fact, these little molecules down here are little acetate molecules. So these are negatively charged molecules from the solvent. And we have this positively charged ring up here. If we then look from the bottom part is really very positive, negatively charged. So clearly these are two different charges, which gives us a bit of an idea about the functionality. I should also say that another suggestion that always comes that this might be tunnel, but looking at the surface representation, we also see that this tunnel is really closed. And even if there was a tunnel, it's actually very, very small. So again, the structure tells us that it might look like a tunnel, but it clearly is not a tunnel. So what else can it be? Well, at this point, we use a bit of crystallography. And we immediately recognize that our 5-fold symmetry of our molecule fits to the 5-fold symmetry of the icosahedra of the virus particle. So if we vaguely remember our virus 101, we remember that this virus particle has 3-fold axis, 5-fold axis, and 2-fold axis up here. And it just happens to be that this five-fold symmetry seems to fit quite well. So what we can do then is we basically take an homology model of our virus capsid, right, up here. So this is an homology model of the virus capsid, done with Swiss model quite some time ago. But I should say an AlphaFold model will not look any different. And we can stick our molecule on top of that. And this is a really very crude model that we have just put on top to fit the five-fold symmetry. And it actually fits very nicely that this five-fold symmetry is repeated, right? And that means that then our negatively charged group up here of the phospholipid bilayer can fit to the positively charged of our upper domain. So what we think this happens that is this sits actually as little spikes on the capsid, right? Fits nicely up here with hydrophilic interactions. And then up here, we have our positively charged part of the molecule that actually then interact with the phospholipid bilayer, with the phosphate groups of the phospholipid bilayer, and destabilizes the host membrane. So this is basically an escape mechanism that does not rely on any catalytic activity, but it relies on the changing of the proton motor force of the phospholipid. So again, it's a nice example that the structure really determines function, and the structure determination enables us to come up with this new mechanism for lytic enzymes. The second example, you've already seen that this is something that we sort of discovered while we were doing the metagenomic sequencing. We discovered that there's a large number of extremophilic so-called candidate phyla radiation bacteria. And these candidate phyla radiation bacteria were only discovered in the last 20 years or so by metagenomic sequencing. This is a phylogenetic tree from this paper where you've got this huge number of bacteria up here called candidate phyla radiation that are genetically distinct from, well, the non-CPR bacteria and all the archaea down here. And it's now been suggested that these CPR bacteria comprise of up to 50% of all bacterial diversity. So although we were looking mainly for viral genes, we found a large number of these CPR genes in our database later. And that means that we sort of found a different additional genetic diversity, where that is termed genetic dark matter. And the point again is that all of these have evolutionary are quite distinct to sort of our normal bacteria that can be grown in the lab, and thus these might have new functionalities. So we basically added some of these bacteria into our pipeline, some of these genes into our pipeline. And what we did is we looked at genes that are highly conserved within this candidate phyla radiation, but have no sequence identity to anything in our normal databases. So this is a protein that we looked at called CPRC4, and you might recognize this fold because I've already shown this in the previous talk. So again, an homodimeric protein, which happened to be a couple of zinc sites, which in this case, again, pre-AlphaFold or just when AlphaFold came out, was used for phasing using MAD methods, right? Now, so from the structure, we now try to find the functionality of this protein. And first of all, we looked at the zinc site, and zinc sites are often catalytic sites, but just the coordination of the zinc with three aspartates points to a zinc site that is rather a structure site and not an enzymatic or catalytic site, right? We were able to solve the structure without zinc as well, and it turned out that even this site, even though this is a structure site, the protein adopts exactly the same conformation with and without the zinc. So again, the zinc seems to be relevant for stability, but it doesn't seem to be relevant for functionality. So then what we did is we looked at the structure and looked at similar structures in the database. And what we found was a really big surprise. We found that this structure was actually structurally similar to so-called VASH proteins. These are human carboxypeptidases that cleave important for cell skeleton. So in blue, you can see our structure, and in green, you see the two VASH proteins. And it doesn't look that they're actually very similar, but the basic fold of the helixes, if you look at these helixes up here, 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 and here, and these two beta sheets. So the basic fold is actually the same. So what this structure comparison tells us that these two proteins are either evolutionarily related or they're structure related from a convergent evolution, right? So the biological function must be different because obviously the VASH protein substrate doesn't exist in CPR mechanisms. The catalytic triad and the enzymatic activity is actually really the same. So they share the same unusual catalytic triad with the carboxy group from a lysine, histidine up to cysteine. So this is the superposition of the active site residues of this human VASH protein with our really bizarre extremophilic CPRC4 protein. So if you've seen the overall structures look somewhat similar, the basic fold of the catalytic domain is the same, but in terms of the active site, the superposition is really remarkably similar. So this then proved for us that this is carboxypeptidase, and we did the biochemical analysis as well. But we still don't know, of course, what the function of this carboxypeptidase in our CPR organism is. Now, why is this relevant in the age of AlphaFold? I think this is extremely relevant because it shows that structures are much, much more similar than sequences. So these two sequences have really diverged beyond recognition, or they have converged to having the same structure from different starting points, right? And that tells us that the structural universe is much smaller than the genetic universe. So again, it's those connections between sequences that have diverged beyond recognition, but still share the same structure that actually machine learning programs can exploit and make the machine learning programs so much better. And one of the features that we all expect in structural biology is that the structure prediction programs are getting better and better every time that there's new structures coming into the database, every time our structures are becoming better and have better resolution. Okay, from that, now I'm going more towards the biotechnological applications. So our biotech companies were in particular interested in these amidases, ultra-stable phage amidases, because they have huge potential in biotechnology, fermentation, food technology, but they're also looked at as novel antimicrobial agents, in particular for bacteria that have highly drug resistant. So what people are trying to do is use these very specific amidases, fuse them together, and make very specific new antimicrobials for particular bacterial pathogens. And again, there's the questions I haven't discussed with Paul. Maybe you can think about this already. Is there some potential to look into antiparasites as well? Now, what we did in this case, we actually used our sequence of information about so-called type 3 amidases, zinc-dependent enzymes that cleave exactly the linkage between the sugar up here up at the very top and our peptidoglycan, right? So we use those sequences to find new sequences that are likely to have the same activity. And in fact, in this can be looked at the shortest of those sequences. So the enzyme that we identified was the shortest type 3 amidase in the entire virus X database. And so we made the protein and in particular looked at thermostability and thermal aggregation with thermal shift assays. And we looked at the enzymatic activity measured by sort of standard cell lyse assays. And these are the activities. So these are the activities, relative activities measured at 60 degrees after incubation for 30 minutes. And what we find that basically incubating this enzyme even up to temperatures of boiling water, or this one up here is autoclaving for a half an hour, doesn't really change the activity. So basically this enzyme is a complete rock. You can autoclave it for 120 degrees at high pressures, dissolve it again at 60 degrees, and the protein has basically almost the same activity. So there's almost the same 100% activity, right? And even if we incubate our AMIP for 6 hours, this is incubation times at boiling temperature. So even incubating for 6 hours retains about 88% of its activity. So in terms of activity, this is certainly one of the most active or most stable proteins that I've ever come across, right? The melting temperature even exceeds the temperature of boiling water, so you can basically get a melting temperature of just about above 100 degrees. So looking at the structure, basically then was a goal to see, do we find structural reasons why this protein is so hyperthermostable, right? Now, again, this structure was solved by SAT phasing, again, before AlphaFold came in, was available, or just about when it became available. And it really is the same sort of alpha-beta hydrolase fold. So in terms of overall fold, it looks pretty much exactly as all the other AMP proteins or the type 3 amidases that were solved with an active site on one side with a zinc up here. So looking at the reasons for thermostability, what we find is sort of the same reasons that other people have seen, and we have seen many times before for thermostability. So there's not one single mechanism that makes it hypothermostable, but it's a number of mechanisms that have evolved over time. So one is that's a really compact structure. There's absolutely no cavities up there. So if you look at this, it's just a little ball of structure. All of the loops are shortened compared to other structures, and all of the termini are also very short and not hanging out. So basically, everything is sort of tied together and kept together. And in terms of amino acid composition, there's a reduced number of thermolabile residues such as cysteines and asparaginins, so residues that might be cleaved or might degrade. So again, not a single point, but usual points in terms of thermostability. Okay, the last example that I have, novel viral recombinases for biotech. And these are basically single-stranded DNA binding proteins, and they're essentially used by bacteria in a wide range of DNA repair and maintenance mechanisms. They seem to be ubiquitous. It can be found in phages, in bacteria, and eukaryotes, and they are called UVSX for phages, RAC-As for bacteria, and RUT59 for eukaryotes. And for example, RAC-As are already widely used in a wide range of in vitro molecular biology techniques whenever we're involving the manipulation of DNA. And often these are used in the kits of manufacturers, and we might even not know that they're being used in these kits. So when we buy a kit for certain, I don't know, plasmid preps or a recombination, then some of these proteins are used as auxiliary proteins because they improve basically those kits. And biotech companies are interested in proteins that work for a particular application. So rather than having one RAC-A for all of the different assays, they would like to have one that works at a certain temperature, one that works at a certain temperature, at a certain pH, and so on and so on and so on. So basically what we did is we just looked at all of the homologs in the database. We found more than 150 candidates and selected these two, UVSXT and UVSXP, out for further analysis, right? So the first thing we do is we always look at thermostability. This is our thermoshift screen that was developed in Durham, which gives you an idea about how the protein behaves at different pHs. A1 and A2 is always the thermostability under under the normal buffer conditions. So 61 and 61 degrees is not super thermostable. Remember, I mean, P can be heated up to boiling point, but it's relatively stable. And then low pHs are in BC, high pHs up here. So these proteins, or these, are both quite stable at lower pHs, which is not a surprise because they come out of these highly acidic lakes in Iceland, right? Both bind single-stranded DNA as expected with binding constants of nanomolar activity. And next one up here. We also then determine the crystal structures. In this case, we actually use molecular placement and we use the AlphaFold models. And looking at the structures, these actually look pretty much like expected RAC-A structures. I just superimposed them both, right? And I also superimposed the T4 UVSX, the closest homolog, in orange. And what we can see is that overall, the structures are quite highly conserved up here in the core of the protein, but then there are larger differences up here in one of the loop regions and also in the intertermini. And I would say this is exactly what we would find with any AlphaFold models. All of these would be highly conserved, and we would say, okay, these look pretty much the same, but then it's the termini up here and the loop regions up here where you have larger differences. And of course, those differences are then responsible for exactly how DNA is bound. Now, obviously, we were interested into biotech applications, and we worked with ArcticZyme together and with Paul Denny together because we are exploring these enzymes as auxiliary proteins for the LAMP assays with Paul Denny. So this is where these two projects actually came really nicely together. We had all of these proteins. We started on these LAMP assays, and again with Paul Denny together, we are now trying to use these and improve the selectivity and the specificity of LAMP assays. And the reason for using those rather than the existing REC proteins is that these are probably much more thermostable, which means that they are hopefully more amenable to any field applications in the future. Now, what's next for the project itself? Overall, we have solved about 25 structures out for 50 million potential genes. So obviously, we can't go on like this. And one of the things we do is we look at cryo-EM structures. Obviously, cryo-electron microscopy has undergone a revolution over the last 10 years. So for those proteins that do not crystallize, we actually have now started with cryo-electron microscopy. And I'm not allowed to tell you what this protein is, but this is, in fact, our first cryo-EM structure of DERM altogether. And of course, we use AlphaFold and any other programs to predict structures and select the weird and the wonderful. So we have started basically taking a subset of sequences, run them all through AlphaFold, and then we use a program FoldSeq that finds similar structures. And this is just one of the examples that what I would call a weird and wonderful structure. Again, the AlphaFold model looks quite convincing up here. Some domains, these domains are certainly correct. Probably these N-termini are probably not correct and predicted with low confidence level. But the problem, of course, is that this is a wonderful structure, and AlphaFold can look at this, but this structure really doesn't tell us anything about its functions. So we still have the challenge to select those that have functions that we are interested in, rather than just selecting those that look some weird. And this is, of course, an ongoing project, and with 50 million potential genes, I'm sure that this will keep me busy for the rest of my scientific career. There we go. Predictive structures is a very limited functional insights, and we come back to what I've said before, and I'm sure Paul echoes again and again. We still need far more experiments, and the experimental validation remains the key. With that, I did put a picture of my group because this is more the Virus X project. So this is my team, particularly thinking Steffi up here, who is leading the lab and did most of the work. And this is the entire Virus X team on our first meeting when we met in Brussels pre-Brexit, and we're all in good mood. So thanks Horizon 2020 for the funding, and these are the funders for that allow a bit of ongoing work with this after Brexit. The CCDC, Cambridge Structured Database, and the European Circular Radiation Facility, and MOSMED. Yeah, and with this, thanks again for having me. Any questions, please?
Okay, so any questions to Professor M.K. Paul?
Okay, thank you very much, Professor MK, for your talk. With your permission, should we take some questions for you? Okay, so any questions from the in-person audience? Okay, so are there any questions in the chat box from our virtual audience? So we see many compliments for your very nice presentation. And one person is saying with many 3 hearts actually, well explained. Okay, so they also want to, they're also asking if they can possibly get the presentations of both of your talks and also talk of Professor Paul, if it's possible. we can share it to the email to these participants.
Yes, of course we can do that.
Okay, so basically there are many compliments for both of your for Professor Paul and for Professor MK for your talks.
Can I ask? Sorry,
Yes.
can I ask Paul if there are parasite phages phages for the parasites? Do you know anything about that?
Yes, there are.
Okay.
No, I need microphone first.
Microphone level, please.
Thank you. Yes, there are. About, I guess 10 years ago, a virus, a corporate virus of a eukaryote was described in Leishmania brasiliensis and associated with heightened virulence. And more recently, a similar virus has been found in one of the old world species, I think Leishmania infantile. So whether that's associated with virulence, I don't think anyone's looked.
Okay, thank you very much. So there is one question from Mr. Rocco for Professor MK. He's saying that considering the vast diversity of viral enzymes, how do you prioritize which viral proteins to study structurally for potential drug targets for biotechnological applications?
So there's two answers to that. So first of all, I mean, these viruses itself are not pathogens for humans. So we really didn't look for drug targets for viruses. So what we did is we looked for targets that can be used for biotech applications, and in particular these recombinases, REC proteins at the very end, or they're called single-stranded DNA binding proteins. They were selected because our biotech partners really use those in their kits. So there we had a sequence to start with. For those sequences that have no similarity and we had no idea what they do, we basically looked at the ones that are widely distributed. So a good example is this CPRC4, which again has absolutely no sequence identity to anything. There's no function associated to that. Any even an AlphaFold prediction wouldn't tell you much about its function. So we basically scored them according to abundance. So this is something that we've the C4 is one of those that we found again and again. And I should say we always had a triage process that we started with, again, maybe a couple of hundred of potential targets, which we then were triaged down to about 10 to 12 targets that went into the experimental validation. So the C4 structure is C4 because it's the fourth out of 12 proteins that the PhD student purified and characterized. And in fact, it's the only one out of the 12 that crystallized.
Okay, thank you very much. Thank you again for your time, Professor MK, and also Professor Dr. Paul. Sir, with your permission, should we go to the closing? Sir, please, I request you, Professor Dr. Iqbal Chaudhry, for your word of thanks to our speaker, probably, and also for the distribution of shields and certificates.
Thank you very much. Thank you very much, everyone, for being part of this very exciting seminar. And we have seen that this was a feast of wonderful three presentations by leading scientists in the field of neglected diseases, and the one who built a team of young and experienced scientists in 6 different countries. We've also seen a picture which presented by Dr. Paul that the team has run a very prestigious medical research council award for best research team. And you could see me as well, somewhere in between. But this credit goes to Paul and this wonderful coordination with everyone. I'm sure that you have benefited from the presentation, and I'm sure that you will be able to keep contact with them. They are very eager to help younger people. And we're so fortunate that we have many young scientists today with us. So let me thank you all, and thank you, Paul and MK. Thank you very much for wonderful presentation. We are looking forward to have you, MK, again with Paul anytime soon, before Islamabad gets very hot. So please do visit us very quickly. Karachi is already inhabitable, so don't worry about that.
I will.
Yeah, so thank you everyone, and then let me invite you to appreciate both speakers with
Okay, thank you very much, sir. Now I would request you to please present a shield to Professor Dr. Paul Denny and the certificate to him. Professor Paul, please receive your shield.
So this is walnut wood, and this is specially handcrafted.
Please appreciate the speaker, Professor Paul. And
For any case, certainly we send to him or will wait for his visit.
Certificate for Professor M.K. Paul is also being given to Professor Dr. Paul Denny. He will take it with him for Professor M.K. Now, I would request our speaker, Professor Dr. Paul Denny and Professor Dr. Iqbal Chaudhary, coordinator at comstech, to present a certificate of participation to our participant, Dr. Hassan Arooj. You really don't need a certificate, but it's just.
I think we need to be quick.
Yes, Dr. Bushra Arshad. Mr. Ahmed Al-Sheikh Yusuf from who has joined us from Rawalpindi Medical, from Palestine, of course. Yeah. Mr. Milad Alvin, he is also a recipient of comstech's scholarship as well, studying at Mr. Tasir.
I'm sure that you're from Palestine.
Yes, yes. Miss Noor, she's also from Palestine. Miss Salsabil, Miss Zainab Noor, Miss Aliza Zainab, please be quick so that we can go to lunch break. Aliza Zainab, Sahar Safdar, Sayyida Rubab Zahra, Sayyida Rubab Zahra, Warisha Sultan, Aruj Fatma, Zara Ishaq, Sayyid Asim Farad, Miss Elsa Javed, Miss Aksa Ashfaq, Miss Anila Javed Sajjad,
You can exchange it here around.
Miss Wafa Hazar, Dr. Ijaz Rafiq, Eram, Tuba, Mudassar Ali Alvi, Shafaq Shakil,
Exchange here.
Asim Zishan, तो मैं दिया मैं आपको तोबा के देखो इसमें शर्जील, सैयदा फातिमा, मलीहा तारिक, please clap for yourself and also for your other classmates. Taskin Kaushal, Tamkinat Mahmood, Hena, Ataur Rahman, Kamal Batul, Taman, Zainab Javed, Sidra Aslam. अच्छा, just one last, yeah. इसके बाद वो भी एक हाथ होगा. This is it. Okay, so ladies and gentlemen, with this we come to end, and I request you all to please come in the front for the group photo.
Ministry of Science and Technology, excuse me.
Okay, please come in
Ministry
the
of
front.
Science and Technology.
अच्छा ठीक है उसके बाद में मैंने बोला था वो आई नहीं. Please come in the front, yeah, for the group photo.
अभी आइए
Dr. Bushra, you can also come. Dr. Samar, आप कहाँ बैठे हैं? आ जाए. Dr. Arun. Now, I request you all participants, please, maybe you can sit in the front if that's possible, because there is no way we can fit into the front. Yes, outside, सर आपको मर्जी है. हाँ, ये भी हो सकता है. So we invite all the local participants to please come in the front to take the group photo with the speaker and His Excellency Professor Iqbal. Kindly नीचे बैठ जाएं जिन लोगों को जगह नहीं बने.